delete, and so on. And then for the V13 to V14 changelog, this is just taking all of the differences between the two schemas and it
Test.
Hey. No. Hey,
Hi,
hey.
buddy.
Can you hear me?
Yep. Can you hear me?
Yeah, okay, I can now. Sweet. Yeah, I started this 5 minutes later after the beginning just so we can catch up.
Sure. Yeah, I mean it's I think it'll kick off when people start showing up. Sometimes it takes a bit for people to get here. Hey, Yo-Yo's here.
Yeah. Yo from 1 a.m., isn't it? In Tokyo?
Tokyo.
Hello. It is indeed 1 a.m., 12 a.m.
Hello.
Oh, the gang's all here. Sharena's here. Alex is here.
Hello. I'm going to be slowly admitting people. Hello, Joscha, Randal. Joscha will never recognize me in this way. Last time we see each other in face-to-face was a year ago.
Good to see you, Sam.
Hey, Randal. Okay, sweet. We've got... for everybody who is kind of just joining, I'm going to be moderating, so most of the tasks I'm going to be doing have to do with logistics. Admitting people and AIs. And we have the meta community on Discord server, also remoting in here, only as observers. Let's see, 18 people, of which 4 are AIs, are yet on the call. Hello, Lucas. Nice to see you.
Hi there as well. Hi, Sharena. Long time no see.
Hello, Lucas.
Indeed, it is a very small world.
Yeah, I think we have people all over the world joining. I might be the only one in Africa, unless someone else here can prove me wrong. So, as the representative of the African continent, joking.
We may have somebody from everywhere except Australia and Antarctica. Okay, a lot of people have joined, so I guess I can just cover the top of the line practical points to do with the call. I wanted to say that everything that happens on this call is tentative, so unofficial. We don't have the time to go into the details. We're gonna take people's words for what they have said. 2 hours seems like a lot of time, but actually it will probably show us that it's not as much as we think. Also, no sacred cows, so everybody just speak your minds. That's kind of what we want to do here. This is the fact-finding mission for whether it's feasible to emulate a fully known neural circuit inside a brain organoid. And it's also our first roundtable, so we have not done others in the past. There will be more to come, more things one-to-one. And I think what I will do, because the first agenda item has to do with the Institute of Sims, is to present today's agenda. I will not spend too much time talking about why we are here. I've done that already individually. So if you can see my screen, I'm presenting. I'm going to present again. Yeah, I'm presenting our agenda document. So I just wanted to say today, we're convening here because Ex Vivo is a R&D program and that is focused on effectively testing the claim that high-resolution emulations of brain function that we can achieve with today's methods can generate some sort of high-growth revenue stream via the drug discovery markets. This call today will not be about drug discovery. It will not be about preclinical testing. It will be about scientific feasibility of emulating a known circuit, ideally in a way that allows us to progressively add modular functionality that we can emulate inside of this culture and observe higher order mental processes taking place in that culture. And I think with that, I will just kind of test run whether we are capable of using these emojis and interacting with each other through the Google Meets capabilities. So if everybody's okay with me proceeding to effectively the agenda item and introducing each of the startups, just give me a thumbs up with the reactions. And I can see them on my side popping up. Okay, that's sweet.
I can actually not see them so clearly as I would like. So I will ask Elliot, the right-hand man for this call, to please let me know if somebody has not given a thumbs up or maybe has a hand up.
We're
I
good
just
to go.
can't see
We're good to go. Everybody's ready.
Yeah. So whenever that happens, just in case you have questions, just jump in, ask them. I'm going to scroll down into our agenda. This document has been shared with you guys, so I can just glance over some of the details. We're going to go into startup presentations. So everybody knows what they're going to be talking about, 5 minutes each, and just ask questions. I'm going to shut up. I'll be a moderator today, not a question maker. And then we're going to hear from Karim about the big pharma landscape for brain-on-chip technologies. This is the biggest commercial kind of input into the call. We're going to hear from Lucy or Yo-Yo about the platform available to our startups incubated in San Francisco. And then we're going to hear from Randal. I'm quite excited about that part too, about auto-associative memory, brain emulation challenge. And then we're going to get into the meat of the matter, discussing with each of you on the call and the individual startups these research questions. So as we go through stage by stage, I'm going to do that and, ask the questions. So first, to begin with, I'd actually like to give the word to Alex from New Brain. I'm going to stop presenting to share with us, your background and what you're doing.
Thank you, guys. It's a delight to open up. Alex K. I am a trained drug discovery professional, you may say. New Brain is a company that is, in a sense, a culmination of many skills that I think I acquired of all of this 30 years in the classic drug discovery. Specifically, we are based on three things. That is ability to manipulate neuronal precursors, number one. Number two, bioengineering, that is ability to put them in an organized biological matrix. And number three is, in a minimally invasive fashion, put them in the specific circuitry in the brain. These are three pillars of a new brain. And that gives us the opportunity to repair, restore, recover, or replace a specific neuronal circuitries. Once again, we're not working with disparate cells. They're well organized. We use our knowledge of embryonic brain development and fairly sophisticated materials from the point of view of biomechanics, bioengineering, as well as interneuronal connection. The whole mission, once again, of the company is restoration of a human brain in the context of very specific disorders. And the ultimate goal, of course, is longevity and organ replacement. Happy to comment, answer any questions, guys.
I don't see any hands popping up. Feel free to just ask a question directly. Here we go, Richie.
Yeah, my question is about characterization. So obviously in the therapeutic context, the characterization is restoration of function. So you put it into somebody with a stroke and they can move their hand when they couldn't, something like that. But from an R&D effort upstream to that, how are you characterizing these? You do this culture in vitro, and then maybe you do animal work or something like that. So
that's exactly right, Richie. So the flow is your standard cell-based biology, you would say. So it's a medium to high content imaging. So essentially we assemble or pre-assemble tiny pieces of this tissue. And on the molecular and cellular level, we make sure that they make sense as compared to control human tissue or mouse tissue specifically. Then the circuitry gets engrafted in animals and we see how the process goes. So it's increasing complexity as we go along. Ex vivo, we like to call it ex vivo, right? Because it's almost a living system. Then it's in vivo, mouse model. Then it's advanced in vivo model. It has to be a non-rodent model as required by a regulatory agency. And then it's human.
Okay, and do you know what degree of circuitry you need prior to implantation? Because this specific application, you're going to get so many cues once it's inside the host that maybe it doesn't have to be wired up really well in advance.
It's a fantastic, as they like to say, $1 trillion question. How good is good, basically, right? That's what you're asking. So truth of the matter is, I don't think there is anybody who will answer that. but I think that it's a signal that you're catching. The general rule of thumb, at least in neuro that I follow, is that if you can enhance your signal by 25%, you're doing well. A good example is that if you're looking at the function in the mouse or in the human, learning ability, memory consolidation, or ePhys, electrophysiology, you will have a very, very clear marker how to enhance the performance of the brain. So yes, we have those cues, and they can happen on molecular, cellular, behavioral, or anatomical levels. This is how clinical trials are built, basically.
I'm going to be very excited to hear your thoughts about all of those markers in the research question portion, because essentially we're trying to work out how much structure and function are we necessarily replicating in order to retrieve a function, a mental process from the culture. But for now,
I hope I can live up to your expectations.
And I will just move on to the next one, a little bit linked to New Brain's company, Frontier Bio. Eric or Sam, feel free to present.
Morning, everyone. It'll be me presenting. I'm just going to share my screen. Okay, are you seeing my slides?
Yep.
Very good. Okay, so slight aside from possibly the other presentations you'll see this morning, at Frontier Bio, we're a tissue engineering focused company, but our expertise here is really going to be around maintaining the viability of said neural tissue. So we're focused on blood vessels, vascularization, and tissue-engineered vascular grafts. So here's our flagship clinical product we're developing, which is a tissue-engineered vascular graft, which we've now progressed into large animal studies. And this has shown excellent patency in a short-term implantation study. But our capabilities that will relate to this project are more around biofabrication of blood vessels and vasculature generally. So we have expertise in biomaterial scaffold fabrication and in making tissue-engineered blood vessels in vitro using degradable polymer scaffolds and biomimetic bioreactor culture. Now, we do have some experience with linking this with neuronal tissue. We have a blood-brain barrier on a chip device that's a microfluidic-based platform where we have vascular endothelial cells in direct contact with neuronal tissue to mimic the blood-brain barrier. And we've used that in various studies, including for traumatic brain injury and for looking at cell therapies. Additionally, we have a lot of expertise in 3D bioprinting and tissue self-assembly. We have our own in-house custom-made 3D bioprinter, and we've used that to produce spatially directed networks of early vasculature self-assembled from endothelial cells and pericytes, as you can see in the bottom right images. So for us, we're really looking at how we can build these vascular networks that will be able to support a dense mass of neuronal tissue to enable the sort of functions that we want to achieve with this project. So I'll now take your questions.
Very nice. For the sake of time, the first hand up gets to ask a question, and then we'll move on. Adrian.
Yeah, thanks a lot for the presentation, Sam. You're showing a couple of different strategies. One of them is let's say, more synthetic with 3D bioprinting, and then the other one is purely cellular with sort of self-assembly. So what's your strategy in terms of which samples will, or what scale of samples would go with self-assembly versus like the engineered approach? How do you deal with that?
It's a good question, actually. We're actually developing a method that can combine the two. So the self-assembly approach really works for the very small vasculature where printing can't get down to those kind of levels. But we do have to have some unity to that larger vasculature that enables us to really interface with it. You know, if we want to connect to a a flow system, we need something that we can sort of plug into. So we're actually working on a method that we can combine those two. So it combines our more classical tissue engineered vascular graft technology with the bioprinting and self-assembly technology. So we think we can actually bridge both.
That's very interesting. Can you say more about about that interface, how you make that happen?
So we use we produce a tissue engineered vascular graft in the more traditional polymer scaffold based method. And then we're working on ways of driving the natural vasculogenesis and angiogenesis processes in a 3D matrix where we've then printed our self-assembled bioprinted vasculature. And we're looking at combining those two, so our natural vessel will branch into those smaller self-assembled capillary networks.
Okay, thank you.
Lucy, go ahead, please. If we run out of time, I'm going to blame you.
Yeah, just briefly, is that with your custom printer or is that with commercial bioprinter like the Formlabs, the scaffold?
So we use our custom bioprinter for that process, but we do have some other printers in house that we use to make materials like bioreactors, for example. So the actual bioreactor you see in the picture, top middle here, was produced on a Formlabs printer.
Gotcha. Thank you.
Sweet. Thank you, Sam, for presenting. We'll get more into depths of it during the open floor and the research questions. Next up is Gleb. Gleb informed me from Duality. He informed me ahead of time that he's traveling in Montenegro. Sent me a beautiful picture from a sailboat. So he might not be immediately available. Are you on the call even? Okay, I don't hear or see Gleb. We're going to move on one step further, and that is going to be Provigen. So Lucas, can you please share what you're doing?
Hi everyone. Let me try to share first. There we go. So to give you guys a short overview of what we've worked on in the past. So this is specifically neurosamples. We have recorded primary Morse cortical neurons on microelectrode arrays. And then the core of what we have been focusing has been representation learning, or more precisely contrastive learning, that I've worked on in Europe at the Helmholtz Center together with collaborators. And so this is one part of the equation on the software side that we have been focusing on. And then the second part of the equation is what we have been conducting with a company, a CRO, also in Germany, back where I'm from. And there we have used novel methods based on active learning to improve over the state of the art in design of experiments. And we have showcased that you can reach actually more than better of the same result that you would traditionally get via design of experiments via active learning. So basically 80% sample reduction, 30% precision increase, precision measured by a reduction in coefficient of variance. And I do know that these kind of are confusing. That's why I'm now reserved more time for the actual question answering part.
All right, anybody has a question at this point? Maybe I will ask one. Oh, Elliot, go ahead.
Yeah, just for clarity's sake, this is around the recording and simulation of neuronal activity, right?
Correct.
Mouse cortical neurons. And so you're kind of recording this as a means of simulating cortical neurons. Is that it?
No, it's actually not simulated. These are real samples that we have conducted in the wet lab. So basically, what these two methods,
the training models are such that you can simulate.
Yes, the training models are essentially not necessarily the simulation, but more identifying what kind of complex nonlinear dynamics are happening on the actual physical data. And the reason why this is useful is because you can interact with like if you see here on the left, you can interact with the samples in real time and based on what kind of interaction you perform. For example, if you apply a certain electrical stimulus, then you can observe if it actually deviates from the normal kind of activity or still stays in the convergence phase, for example. Like basically you can observe if the behavior of the cells changes significantly in comparison to its normal behavior.
Alex, please.
Very quickly, Lucas, the topology and resolution of your arrays. Could you comment on that?
Yeah, so we used the microelectrode arrays with about 30 microns of diameter of P.PSS, so quite closely actually to what this project will be about. And also the resolution was 60 channels in particular.
It's essentially a single cell resolution. Gotcha.
Not really single cell. I would say more like 3 to 5 cell resolution
In
on
between.
average.
Okay.
Exactly.
Thank you.
Yeah, one of the, just to add a little bit of glue between Provigen and the rest of the companies, something that if there is sufficient hardware to support it, Provigen allows, I suppose, if I can put it in these words, of a control of both the protocols during morphogenesis and during function entrainment.
Correct.
So if you identify a specific circuit, Provigen's software will execute the command over the hardware to entrain a particular function of that circuit in the culture. And I'm glad that you're focusing in your presentation more on the computational nonlinear dynamics problem and its measurement, because today, Gleb, if he's not on the call, and neither is another shortlisted participant, Thomas, there are two computation-first startups working on the simulations of both synthetic morphogenesis and on the performance of a circuit that we know based on empirical data like a functional connectome, electron microscopy connectome. And these two companies have not showed up today. Unfortunately, they were unavailable. So Lucas, and I think alongside Randal, will be representing more of that side of things in our efforts to emulate a neural circuit in a culture. So last, I think it is last but not least, yes, we have Nexi. And the reason why I left Case to be the closing presenter is because of the unique brain-on-chip proposition for drug discovery that is core to Nexi's mission. So if you don't mind, Case, please go ahead.
Hey everybody. Thanks, Sam. Yeah, so I think I know some of you. Eric, good to see you again. It's been a while. Karim, good to see you. Yeah, so I'll just quickly kind of go over what we're building without too much detail. We are using high-density microelectrode arrays and microfabricated architectures to basically reconstruct neural circuits that are reproducible, that are confined, that we can then either look at single cell resolution in 2D with iPSC or ex vivo dissociated cultures, or we can use 3D electrode arrays to peer within the activity inside of a tissue construct like an organoid. Because of the limited range of cells that we can produce with iPSCs, while we can get single cell activity resolution, which is very valuable for things like biocomputing and some of these applications that we're talking about, entrainment, the range of brain regions that we can generate with organoids gives us the ability to create really sophisticated circuits like the cortical striatothalamic circuit, things that Sergio Pathk has been doing, but we're going to do it scalably in a way that is amenable for drug discovery. So that's kind of the, I have some images I can share, but with the amount of time we have, I don't know which ones would be most valuable. I have some of the work that I've done previously. Using this type of paradigm, we produced the first IND approval without animal testing by showing efficacy data to bypass animals. And so the proof of concept is there. A lot of that work led to, well, we suspect it wasn't cited in the FDA Modernization Act, but it was immediately following the publication of this. It was acquired by Sanofi. It's now in phase 3. It was the first, and so we suspect it was pretty instrumental in getting that FDA approval, that legislation. So I'll open up for questions with that being said.
Yeah, if there is no questions, let's go on to Zachary. Go ahead.
Yeah, so maybe a question in the opposite direction that you'd expect, but what are some of the simpler circuits or like neural counts that you're able to reproduce consistently?
One of the things that most people have been doing is just using cortical circuits and trying to entrain those into something that they can expect for things like excitatory and inhibitory balance or long-term potentiation. Those are probably the most canonically developed circuit readouts. And that's what I've done some work like that in the past. LTP induction, you can use high frequency stimulation as one, or you can then induce LTP. You can use pharmacological agents to do that as well, to modulate it, like AMPA and NMDA modulators. That's probably the number one for like a memory formation circuit type of model. Excitatory inhibitory is really relevant for seizure-genic compound testing.
Sorry, I'm on public internet here, so I dropped off for a second, but I think I got most of the answers. Thank you.
Yeah, I'm really interested in seeing if we can create more elegant structures for deeper regions of the brain that are not quite as, I would say not simple, but straightforward. That's where I think there's a lot of disease modeling potential.
Mm-hmm. Research question number one asks, what kind of circuits can we emulate in a culture, right? At what level of complexity and at what scale of parallel processing, of parallel simulation, emulation? So when we get there, I'm sure we're going to have a fun time exploring this question of Zach's. So next up to talk would be Karim, who's already on the call. Can you shed a bit more light about what does the medtech or big pharma in general think about preclinical testing and how perhaps having a functional emulation of a known circuit would help accelerate that and reach their market? Go ahead.
Yeah, thanks, Sam. So medtech, neurotech has several aspects, but when you look at the landscape, the M&A landscape in the past few years, it has been dead most likely, but again start early this year it start strong with the deal from Axonics. So we can here to organize it, we have medtech, diagnostics, deep tech, and all of that in neurotech, and we have pharmaceutical compounds. So for why it matters to strategics and M&A landscape, when you look at it right now, you can see there's Boston Scientific acquisition of Axonics. And also when you, on the pharmaceutical, you can see 2 weeks ago with AbbVie and Gilgamesh, 1.2 billion for their compound. So, and what are the strategic want and when to engage with them and what to do, what not to do. So what they want is they want clinical evidence is is this working, right? For medtech, what are the proof of concept, right? With regulatory clarity, what's your 510K? Is it PMA? You have this mapped out or not? And if you have some reimbursement, are you gonna piggyback on old codes or you need new codes? So you need to see some clear roadmap. For pharmaceutical, they need the proof of concept and they need okay, what assets, how are you gonna designate this? Is it gonna be fast approval or do you have the IP or how this is going to fit within our portfolio of compounds? Oh, I see Sharena has a question.
Yeah, this seems like there's a lot of potential research infrastructure that is being built here that can expedite these larger companies' paths towards approval of drugs and other compounds. So where does that play into this, given that the things in the mind ex vivo don't seem to be trying to be reimbursed for directly?
Yeah, great question. So the strategic don't expect you to have everything ironed out. They wanted to see as early as proof of concept, and that one is the best part or the sweet spot to engage with them. They don't want to see you have all of your infrastructure built, because if you have this, you will go through an IPO. You don't need the big strategic, right? So you want to see phase 1A, phase 1B. That's a sweet spot when you actually start to engage with big pharmaceutical, and that's their sweet spot too, because your valuation is high enough but not too high. for them to pay. And the other things they look at, like, okay, is your compound or your medical devices are going to be like fit within our infrastructure? Can we manufacture this easy or not? Because you don't want to build like a new factories and new like labs for this specifically. They wanted to utilize what they have to already like use it. And that like get us to what to do, what not to do. Like first, like, put your ducks in order, like, have like trial endpoints and like have like all of the things mapped in the team, like, like everything, like, from, and then don't have like, like only one strategic. Don't get into exclusivity with one strategic. Have a multiple better, which is like, it will increase your valuation, but also like, don't put your eggs in one basket because they can be like, unplug it and they just don't care. Third is like, don't ignore like compliance, quality for like speed. If you do this, yes, you will finish faster, but at the same time, you will have a bad product and they are not gonna care. The deal will follow suit. Yeah, and like have your IP like clear, have your regulatory pathway clear. Doesn't mean you need like the actual like regulatory approval. It means like you already like spoke with the FDA, you have a regulatory person in your team, you have a reimbursement person in your team. Yeah, Sam.
Yeah, I have something perhaps meaningful to add about also Sharena's question.
Yeah.
While the first asset of mine, Ex Vivo, might not be under strict regulatory scrutiny, at least at its creation time, there are other, like we mentioned, like we heard from Alex and from NewBrain, there are other companies here whose strategy has to do with introducing these biorealistic cultures into humans, and those are obviously subject to rigorous scrutiny. Neurona TX last year, this time, was valued $650 million, grafting single cells. And that's one company that's in early clinical trials, but there are a lot more companies in this vein. Before I give the word to Alex, I just want to acknowledge that Gleb has joined. And while Karim is already part of the presentation of the Mind Ex Vivo team, and we would be followed by Elliot normally, I'll give the words to Gleb to present briefly what Duality is doing. But first, Alex.
I just want to add, thank you, some more flavor to what Karim and Sharena were saying. There is a huge interest towards bioengineering of tissue and brain in particular, multiple aspects in pharma, make no mistake about that. And I physically spoke with at least probably 12 of them. And whereas the majority of them prefer to follow a standard phase 1B, phase 2 proof of concept in the clinic, there are companies that are willing to help you out at the very earliest stages. Partnerships, out-licensing, co-validation. This is definitely a venue that I would like to explore. And the reason is because you do need an older brother, so to speak, with the clinical access and regulatory expertise at this stage. So there are good, willing partners out there.
Yeah, no, Alex, you brought up an excellent point. So, and most of big pharmaceuticals, when they wanted to acquire, they wanted to see multiple assets, meaning they wanted to see is like, okay, what's your indication, 1, 2, 3, 4, and they will net and peg. And the reason for that is beneficial for two things. Big pharmaceuticals, like they don't want to put their eggs in one basket, but you as a startup, you don't want to put like, you want to sell for the highest bidder as well, right? Or get acquired. So the other thing is like, you have like, all of this like milestones, you can go to different like strategics and partner with them. So for like, like to help you with like the data, to help you with other things, but also as a potential acquirer, so they can like have 2 or 3 acquirer, like, or like strategic on your like bidding for you pretty much.
You guys talking about brain chips?
Okay, thank you, Karim. And this will conclude our kind of commercial segment, commercial go-to-market segment, really. We're going to talk about science and technology more, and I think Gleb is going to tell us about their project at Duality. After that, it's going to be Elliot, right? Yeah, Elliot, and then Lucy, and then we get to research questions. So, Gleb. By the way, while you're pulling up any presentation or joining, we have 35 live streaming scientists in Metamorph Discord. Looking at our conversation right now. All neurotech researchers. So without further ado, Gleb, please go ahead.
Very fun. Good to meet everyone, guys. Okay, so there is very little context to what needs to be presented from what I have engulfed. So please do feel free to stop me by time and redirect the conversation. But I can give you a very holistic overview of what we're up to. Essentially, in a nutshell, we're trying to emulate organisms in a very traditional fashion of essentially step changes. So beginning with C. elegans, at the moment we're working on emulating C. elegans to the highest fidelity possible, and that includes basically everywhere working from software to wetware to hardware. And our very first kind of state-of-the-art organism that we're trying to emulate is a zebrafish. We are currently in the process of, again, predominantly working on seals, but very shortly we're transitioning onto zebrafish. And the key rationale, again, very kind of sparse understanding of who is in the group and who understands the general emulation roadmap, and why zebrafish is kind of the important organism. But to very quickly highlight, it is the very first vertebrate. And of course, if we have the capacity to emulate complex behaviors across different layers, from layer 0 to L1, L2, L3, L4, L5 of behavioral characteristics, so everything from reflexive behaviors to kind of more complex social or goal-directed behaviors, like, for example, predictive coding and hunting, and then onto social behaviors and perhaps even behaviors illicit of identity, then it becomes, I guess, a very good proof of concept of different complexity hierarchies that you can model using emulation approaches. So this is our very first kind of approach, upon which hopefully this will speak for itself, and we will have more capacity, more talent, more capital, and more partnerships to model higher order organisms, i.e. mice, and so on and so forth. But that is at the very beginning objective. I guess we are also discussing with some potentially modeling things in vivo, so things in the petri dishes, and then actually decoding it by essentially using expansion microscopy. But that's something on the sidelines to begin with, and we're predominantly focusing on zebrafish. That's pretty much it.
I was going to ask Randal to give a question, and he beat me to it. Please go ahead.
Yeah, my question is, you said that you're currently doing C. elegans and you're going to be moving to zebrafish. Why are you deciding to make that move now? Do you have C. elegans done, and how do you determine when you've completed a correct emulation of C. elegans?
Yes, very good question. So it is much more of an open exploration than one might actually elicit. We have a set of milestones that we want to accomplish, both in C. elegans and zebrafish. More than happy to send you our kind of roadmap and detailed technical plan.
I'm not hearing diddly. They must be hearing each other through a different mechanism.
Hey, wait, I can't hear nothing. Is it just me? Oh, am I on the wrong one?